Hi, everyone, and welcome to North Star ALS's Illuminate Precision Medicine, the Precision Therapeutics panel. We have our research and industry panel today. We're so very excited to have all of you. We're going to just give a minute for people to have the chance to join. Thanks so much for taking time out of your day to be here. And then we'll go ahead and get started. I'm going to go ahead and share my screen and run through a few housekeeping messages before we dive into our content. And so here we go. All right. Just first up, we received several requests for translation. And so Zoom does have built-in translation capabilities. So we got requests from France and from Italy and from Germany, but there are other options as well. You will need to just find the spot in Zoom to turn it on. And so feel free to do that, but those translations should be available. And I think for the recordings that YouTube should provide translations as well. North Star ALS is the independent science hub for the ALS community. We're here to offer support to ALS families to connect with research opportunities, to contextualize current research findings, and offer insights to researchers straight from ALS families. Today's information is purely for educational purposes. There is no medical advice being offered today. And then just a few housekeeping notes. This webinar is recorded. It should be up on our YouTube platform tomorrow. Thank you so much for your pre-submitted questions. We do keep track of them before every single webinar, and we will try to get to relevant questions today. Feel free to drop questions into the question box, and we'll try to answer as many of those as we can at the end of this panel. Today's panel it's about a 60-minute discussion, and then we'll have an additional 15 minutes of live Q&A. We will be prioritizing questions that are most relevant to today's discussion, and then we'll collect your questions and try to get back to you by email on everything else. But also look for answers to your questions through the additional content that we're sharing through this program. We've had several webinars, we've had social media content, we have podcasts, and so many of the questions were very relevant to one of those specific things. And so some of them have been answered there, and we try to let you know when that happens. We really wanted to give a warm thank you to our sponsors and partners who've allowed this programming. We are so grateful to these organizations who have shown us so much support and support the community in so many ways. And with that, here's our panel for today. We're absolutely over the moon to have them here today. We have Dr. Neil Shneider from Columbia University, Dr. Amy Easton with Target ALS, Dr. Olga Uspenskaya-Cadoz from VectorY, and Dr. Eric Green from Trace Neuroscience. So I'm going to go ahead and stop sharing. But it's been such a pleasure to have this group together. And I think when we were putting this program together, I really wanted to bring this type of panel together so that we can kind of compare and contrast what is truly happening in ALS research today. And that's our goal here. So we'll start with a few introductions and kind of learning at a high level what's being worked on by each group here. Before we do that, Lori Andre, I'm just going to have you introduce yourself real quick as well.
Hi, I'm Lori Andre. I serve as a board member on North Star ALS, and my husband diagnosed with ALS in the fall of 2026.
Thank you so much.
2016. Sorry, 2016.
Thank you so much, Lori. Lori is going to be my co-host today and just help us manage questions and all of that. So our first topic is targeted ALS therapeutics and kind of how did we get here to today? So Eric, I'm going to start with you. UNC13A was identified in 2010 as having some connection to ALS risk. Can you give a brief overview of your program and kind of give us an idea of how you got to developing this ASO that you're working on?
Sure, and just wanted to start by thanking you, Nadia, and Lori for having us here. We're excited to be here with Neil and Amy and Olga and telling you about our work at Trace. And I think UNC13A is a good example of the way that scientists build upon each other's work in order to go from a basic discovery to something that is actually ready to think about translating into a medicine. And the story of UNC13A actually begins more than half a century ago with work that led to a Nobel Prize, where it started out in worms, looking for genes that, when they were mutated, led those worms to become uncoordinated. And this is where the name UNC13A came from. And then over the course of the next several decades, scientists figured out why worms that with this mutation became uncoordinated. And that's because UNC13A is essential for allowing the communication between nerves and muscle cells. And it allows the vesicles that contain neurotransmitters, that are the chemicals that allow nerves and muscle cells to communicate. It allows those vesicles to be formed and to fuse. And this has been known now for decades. And people, as you said, Nadia, knew that UNC13A was associated with ALS, but they didn't understand how. And the key discovery that unlocked this as a potential approach in ALS came from our founders at Trace Neuroscience now about 5 years ago. And what we discovered is that nearly all people with ALS lose the UNC13A protein, this essential protein for communication in the nervous system. And they lose it in a very particular way, which is that the RNA, which contains the information to allow the UNC13A protein to be made, is not properly processed and put together. And when that happens, the RNA is degraded and you don't get the protein. And so that insight really allowed us to think about having a very precise and targeted approach to try and bring back healthy levels of UNC13A. And this is what our therapy does, is it's designed to go and bind directly to that RNA and correct its ability to be processed and to make functional protein. And so this is the approach that we are following with our lead program, TRCN1023, that has entered clinical trials earlier this summer, is that it's able to go and find that UNC13A RNA, ensure that the protein gets properly made, and we hope bring back the proper communication between nerves and muscle cells and slow the development of paralysis in people with ALS.
Thank you so much, Eric. That was wonderful. And Olga, let's go next to you. TDP-43 has been known to be central to ALS biology for so long, and you have a very interesting way of targeting it at VectorY with your current trial. Maybe you want to talk a little bit about your therapeutic and give a quick intro for yourself.
Sure, I will be happy to do that. But again, Eric, I would like to start by thanking you, Nadia, specifically for inviting me. I'm super humbled to be part of this panel. It's great. So I'm part of VectorY Therapeutics. I'm Chief Medical Officer here, and our mission is to develop a pipeline of vectorized antibodies to address neurodegenerative disorders. And our main priority is ALS. In ALS, we are advancing a vectorized antibody called VTX002. It is an investigational therapy designed to address, as you said, one of the most common biological changes seen in ALS, abnormal TDP-43. We know that in healthy motor neurons, TDP-43 is mainly localized in the nucleus, where it helps regulate how genes are read and how RNA is processed. And in most people with ALS, TDP-43 actually moves out from the nucleus, gets mislocalized, and then holds incorrectly and forms harmful clumps in the cytoplasm. So it's not necessarily an aggregation or loss of function in the nucleus. It's really a mislocalization problem. And so what we are trying to achieve with VTX002 is to deliver directly into the motor neurons genetic material which encodes for a small antibody-like protein. And we are using AAV delivery, adeno-associated viral delivery system, in order to get inside the cell, because unfortunately, classical antibodies administered peripherally just can't get where we need them to act. And so then the aim is to recognize the abnormal TDP-43, reduce the harmful aggregates, and support restoration of more normal TDP-43 function inside the nucleus. So obviously, this is still being studied. VTX002 is now in the clinical trial, which we call Pioneer ALS. And of course, we don't know yet whether it will slow the disease progression. Obviously, the scientific hypothesis and the goal is that it will be the case because we are targeting the central disease mechanism and not only treating symptoms. What I think is great is that in addition to VTX002, many companies are advancing other treatment strategies also targeting TDP-43 in different ways, and we will learn one from each other and also benefit from the biomarker development the field is doing currently on the TDP-43 front. So I think it's very exciting times.
Thank you so much, Olga. That was amazing. And Neil, we'll go to you next. So I remember years ago, I've been following things for about 7 years. And years ago, when the FUS ASO was getting started up, there was some discussion and concerns about the safety of targeting the gene with an ASO. Today, that program is approaching completion of a phase 3. We don't quite know how it will read out yet, but you've also now expanded into other rare mutations and genes, and you're working to advance things. So can you give us an idea of some of the genes that you're working on, and what are the pros and maybe some of the challenges to working on these individual mutations with ASOs?
Right. So yeah, as you said, I started this effort now many years ago, 8 years ago, focused on FUS because it was an area of research interest in my laboratory. But I see patients, and these patients came to my doorstep, and we began an effort to develop an antisense therapeutic for FUS ALS, and as you said, that led us to treat a handful of patients through a series of investigator-initiated research INDs, expanded access or compassionate use INDs, as they're sometimes called, and that de-risked the program sufficiently, provided enough, I think, safety and efficacy data to interest Ionis in sponsoring the global phase 3 study, which will read out just in a few months actually from now. We're expecting top-line data as early as October. So that experience for me was really important. I realized that these and tofersen, Qalsody, of course, helped to make this point that this strategy of approaching the disease using gene-based therapeutics, antisense oligonucleotides, and related ASO antisense drugs was really powerful and could be applied to the genetic forms of ALS. And so based on our FUS experience, we started something called Silence ALS, which is an initiative to develop individualized ASOs for rare genetic forms of ALS. And we're doing this in partnership with the Eleanor Foundation, which is a group started by Stanley Crook, the founder and longtime CEO of Ionis, who went on to establish Eleanor with the idea that this, technology that he developed over 3+ decades, could be applied to the very rare, ultra rare, cases of not just ALS, but all genetic diseases. And we seized on this as an opportunity to apply this to the ALS community and to help patients who carried these variants that were so rare that no company was going to invest in this because there were just too few patients to do a trial, too few patients to establish a market that would be profitable enough to, for a commercial entity to make the effort. So we knew we had to do this on a nonprofit basis, and that was the start of Silence ALS. We focused on several genes in our initial efforts, CHCHD10, TAR-DBP, which encodes for TDP-43 itself. But we have several other programs active, and I know you want to talk about this more in detail later. But the idea really is that we need to approach each genetic form of ALS with a therapeutic that makes sense for the biology of that specific form of the disease. We need to tailor our approaches to the individual subtypes of genetic ALS. And we need to do this, I thought, through a platform that produced ASOs that were safe and effective and delivered in a way that could help as many people as possible around the world. And I'll just say finally that although we are focused on the rare genetic forms of the disease, and people often will say to me, why so much money, why so much effort focused on these rare genetic forms of ALS? What about the 90% of cases in which there is no pathogenic variant, no genetic cause? What about them? And I would say that, we are learning so much from the application of this technology to the rare genetic forms of the disease. That was true of tofersen. I think it's going to be is already and will continue to be true of ulefnersen, the FUS ASO. And it's teaching us about what success looks like in ALS, what a successful trial actually looks like, what it looks like for the nervous system to heal itself, and repair, even recover function lost in the course of disease. And some of the genes that we're targeting and the mechanisms that those genes are involved, these mechanisms are clearly relevant to the larger sporadic population. What we're doing in the rare genetic forms of disease will apply in time to the larger sporadic population. And the more we learn about the biology of ALS, the more we learn about these many ALS related genes, their interrelationships, their interactions between the proteins they encode, their common role in pathogenic mechanisms, the more likely I think it's going to be, and the clearer it will be that these gene-based therapies will be seen as broadly applicable to ALS.
Thank you so much for that great explanation. So many great details there. So Amy, we're hearing about different therapeutic approaches for different groups here. And we're so excited to have you. So if you can maybe give us a quick intro to yourself and also let us know how Target ALS works across the research ecosystem to bring together all these different pieces: funding, shared data, biosamples, infrastructure, so that programs like these can move forward. And your organization is Target ALS, and we're talking about targets here, so this is perfect.
Thank you, Nadia, for inviting me today. I'm Amy Easton, VP of Scientific Programs for Target ALS. And prior to joining the organization in 2022, I worked for quite some time, almost 20 years in drug discovery in biotech and pharma. So I really enjoy today bringing my insights to bear on how we're going to fill in the gaps that can't be covered by biotech or pharma and can't be covered by academics, to help bring us all together as one group to accelerate therapeutics and biomarkers for patients. And so we have developed a holistic strategy to do this, and that includes the research that we fund. So, for example, we really focus a lot on bringing together multidisciplinary consortia, which allows academic groups who have specific kinds of expertise and knowledge to work together with biotech or pharma, who obviously have a different set of expertise, to allow this new target identification to develop and to actually be implemented ultimately later in the clinic. So we have had for many years a big emphasis on funding those academic industry consortia to do just that. And we also provide not just our grantees, but any researcher around the world a whole suite of resources and tools that are hard to get otherwise to help accelerate their research. So in particular, some of our resources and tools include postmortem tissue. So Target ALS funds across a collection of 6 different institutions the generation of ALS brain and spinal cords from about more than 600 cases now. So we have one of the largest brain banks for ALS that exists. And the tissue are collected so that we can distribute them to both biotech and academics alike to do the detailed mechanistic work they need to do. But also we generate what we say multimodal data sets. So we're generating a lot of different kinds of data from every single donation to tell us as much as we can to fill in as many pieces of the puzzle of what's the underlying disease biology and how can drug companies best intervene. And so we have this comprehensive resource that is actively utilized by many folks around the world. In addition to postmortem tissue, we also have developed a longitudinal biofluid core. So I can tell you more about that later today when we talk more about biomarkers, but essentially we have again, a very large collection of CSF and blood from patients who've kindly donated their samples over time through our global natural history study. And so we have more than 30,000 samples banked, and we've been distributing those samples for at least 2 years to help accelerate biomarker research for everyone across the industry. And those two pieces are really important for identifying new targets and figuring out how to precisely target them and which patient populations those therapeutics are best suited for. So there's two different pieces that we're really trying to help tackle: developing precision medicines like ASOs or siRNAs to very precisely manipulate the target of interest, whether it's RNA or protein, but then also providing these rich data sets through our data engine out to researchers around the world to mine big clinical data sets to identify specific subsets of patients that might benefit from one treatment over the other. And so one really nice example, just add on since Eric is here today, is work that we funded through a consortium of Trace Neuroscience as they were just getting off the ground, collaborating with Stanford and Michigan University, as well as folks in Germany to really establish kind of the preclinical work and preclinical validation for the UNC13 program, which was then complemented by, again, biofluids to help develop their biomarker strategy. I'll stop there.
Yeah, if you weren't going to mention it, Amy, I was definitely going to jump in and say, yeah, we've benefited from almost every one of the initiatives that I heard you lay out there, from kind of the pre-conception days through to the work we're doing today to develop biomarkers as part of our clinical trials. So it's having a big impact on the work that we do.
Thank you so much, Eric and Amy, for all of that. It's just incredible what you're doing. So we're going to shift to talking a little bit about what's changing in the pipeline and what makes something a precision therapeutic. So Neil, I'm going to go to you first. Maybe you can tell us a little bit about how you create these therapeutics for these different mutations. What's the process involved? That you might want to unmute.
Yeah, that would be helpful. So, our efforts are focused on the most upstream proximal steps in the pathogenesis of these rare genetic forms of ALS. And in this, we're sort of, similar to the VectorY program in that the vectorized antibody that Olga told you about is going after the abnormal protein aggregate of TDP-43 that is many believe to be the driver of neurodegeneration in ALS and other TDP-43 disorders. So similarly, we're going after those most upstream events in ALS, genetic ALS cases, and these are in people who have variants in genes. So it can be just as in the case of tofersen, it was SOD1. For the FUS related cases, we developed jacefusin or ulefnersen, and there is similarly in all of our genetic cases a target, a specific target of a gene in which the variant, the ALS causing variant, is found. So, what do we go after? We go after the disorders, first of all, that present themselves to us, right? We're very reactive in that way. We spend a lot of time through Silence ALS and related programs focused on pre-symptomatic carriers. One that we have at Columbia called the ALS Families Project, but now sponsored nationally through the Prevent Consortium. We identify pre-symptomatic carriers. We work with our colleagues to identify symptomatic carriers of these pathogenic variants, and then we decide whether or not we have an approach. So we go after with ASOs, which is the modality of choice so far for Silence ALS, mechanisms that are amenable to an ASO. And that really involves a mechanism that we call dominant or gain of function mechanisms, where there's a protein made that we think is causing the problem. And the idea is, at least in our early efforts, to eliminate that protein or reduce the levels of that toxic protein in order to restore balance in the cell, protect cells that are not yet affected, help cells that are affected to recover structure and function. And so we spend a lot of time thinking about the biology of the disease specific to each of the genetic forms of the disease. Do we understand that biology? If not, we've done experiments to try to clarify this is part of the preclinical Silence ALS program, and then we decide on a strategy to move forward and begin development of the ASO. So there are examples, in the case of CHCHD10, which is a cause of ALS, very rare. It's not just a cause of ALS, but other proteinopathies, including frontotemporal dementia, which is a sibling of ALS. We didn't know enough about CHCHD10. We were afraid initially of targeting it in a non-allele specific way, meaning knocking it down entirely. So we worked with mice and cellular systems to convince ourselves that the loss of CHCHD10 was tolerable in mouse and cells. And on the basis of that, we went forward with a non-allele selective ASO, which we then brought into the clinic. Very similar story to what we did with FUS, right? And before we went forward with FUS ASO that was non-allele selective, we did a lot of work, years of work, to convince ourselves that the loss of function was tolerated and that we weren't going to hurt people with an ASO that non-selectively targeted the FUS RNA. And so this is what we do in each case. In CHCHD10, we decided to go forward with a non-allele selective approach. For TAR-DBP, there's much more concern there about knocking TDP-43 down too much. And so we're, at least initially, going after this with allele selective ASOs, going after the bad copy, but leaving the good copy alone. And doing this in a way that allows us to study the system as we move forward, dose escalating over an increasingly higher dose of ASOs, monitoring the degree of TDP-43 knockdown, using markers similar to what VectorY, I'm sure, and other groups targeting TDP-43 will be using, based again on the biomarkers that Amy and Eric were referring to. So again, a lot of what we do is based on a knowledge of the biology of ALS, common to all forms of the disease, but also unique to the individual rare genetic forms of the disease. And part of the precision of precision medicine is understanding that biology. And so we need to tailor our approaches, and the precision comes at the level of the gene, at the level of the variant, at the level of the haplotype, the genetic makeup of the individual, and it relies really heavily on, again, an understanding of the biology of the disorder.
Thank you, Neil. You're working across so many different types of ALS, so it's a big question that you had to answer, for sure. So Amy, from your vantage point across the ALS ecosystem, are you starting to see some shift towards talk about precision therapeutics? I think this panel, again, is a very good representation, but do you think that shift is starting?
Thanks, Nadia. So I would say historically, Target ALS has tended to fund projects that were already grounded in some form of specific RNA or DNA finding. And so we happen to have in our portfolio quite a number of projects that are already considered precision medicine approaches. Having said that, I think there are broadly across the ALS field, many groups, biotech and academia alike, who are really trying to dig into some of the robust human clinical data sets that we've generated, and we're sharing out to identify new targets, whether it's at the level of the DNA or RNA or protein, to specifically modulate them. So I think today in the short term, we are just at the tip of the iceberg, really, of doing some precision medicine. We're seeing some of that there. Some of the will still be more classic, if you will, like the example of tofersen, where you've identified an autosomal dominant mutation that's causing the disease, and you're using a therapeutic to reduce that mutant gene. And so there's still quite a bit in that space, particularly when it comes to thinking about C9 or 72 patients and how to tackle their disease using CRISPR-based approaches, for example. But we are going to see a lot more, I suspect, over the coming years, a couple of years even, where new biotech are going to focus in on additional new subgroups of patients who have common similar biological alterations, for example, changes in neuroinflammation or changes in the peripheral adaptive immune system.
Thank you so much, Amy. I think that's a really good segue into Olga and Eric and your programs. I think what I love here is, again, the contrast between Dr. Shneider, who's targeting these individual rare mutations, and Olga and Eric, your therapeutics are targeting very broadly, but they're still considered precision therapeutics. So maybe, Olga, we'll start with you if you want to talk a little bit about how this is, how your therapeutic is still a precision therapeutic, even though it's targeting broadly.
Yeah, so I think that precision medicine in general is a very fancy term, which can mean everything and nothing. But for me, essentially, it is the question is, how close can you get to the root cause of the disease? And this is why I would explain precision medicine or precision therapeutic is something which is based on the biology that drives the disease. And it doesn't necessarily need to be only for a person who has a rare inherited mutation. We just, in some cases, we can get as close as, targeting the disease-causing mutations. But in some cases, we have to satisfy ourselves by the biological pathways we understand well enough, and in case they're druggable. So some precision medicines are designed for one gene or even for one person, and that's great. But others may be precise because they target a specific disease process that is shared across many patients. And TDP-43 pathology is seen at the target in the majority of people living with ALS, and not, it is a broad patient population, but it is still a precision therapeutic. And so this is why we hope that VTX002 is designed to be mechanistically precise, because it is aimed at abnormal TDP-43 biology, and it is potentially relevant to a broad group of patients. So for me, precision does not always mean small patient population. It means that the treatment is guided by the understanding of the underlying disease mechanism.
It's great, Olga. Eric, what would you have to add about your therapeutic?
Yeah, I mean, just I think to echo some of what we heard, I think we're fortunate in ALS that the era of precision medicine in ALS is here, and I think tofersen really heralded that. And the question is, how do we bring that era to the rest of the people with ALS? And I think it starts with developing medicines where we have a very clear idea of what the mechanism of disease pathology is, and then designing those mechanisms to intervene very specifically on that pathology that we've identified. And this is the foundation for the work that we're doing in UNC13A, where that first understanding that what goes wrong is the processing of the RNA, and then taking advantage of a modality, which is an antisense oligonucleotide that can be designed to specifically hone in on those RNAs and allow for correction of that missplicing. And so that combination of a precise understanding of the disease pathology with a therapeutic modality that can precisely target that pathology. So I think that's one key component of it. And then the other thing that I'll add is that in the rare populations with mutations in SOD1 or mutations in FUS, I think we already have an understanding of how we can select the patients who are going to be most likely to benefit from therapies. I think that challenge is one that's still important as we think about that broader population of 97%, where it may, although there's maybe common mechanisms such as dysfunction of TDP-43, there may also be important subsegments within that population. And this is something that we're particularly interested in our clinical trials at Trace, is understanding how we may be able to better predict which patients are going to be most responsive, taking advantage, for example, of things that we understand about genetic variation in the UNC13A gene that could help us to identify those who are going to be most responsive to our therapies. And this is something we'll be testing as part of our clinical trials.
Thank you so much, Eric. And I think that actually segues really well into our next, our bridge topic here. I'd love to talk a little bit about the path to a clinical trial, which can look really different. This is something that came up in the planning for this particular session, that the path to an individualized ASO versus the therapeutics that are targeting more broadly, even just to get it to phase 1 might look a little different. So Eric, maybe you have some comments on that, on what it took to get your drug to trial.
So, I mentioned the experience we had where was about 5 years ago that we had this key kind of unlocking insight about what we thought the mechanism was by which UNC13A was linked to ALS. And that suggested to us this therapeutic approach of developing an antisense oligonucleotide. And what we've really been focused on over now the past 5 years is how do we go from that general idea of having an antisense oligonucleotide that is effective in a cell, in a dish, at being able to correct the improper splicing of UNC13A, to actually having something that we all feel comfortable that we're ready to start giving to patients. And this involves us having a really good understanding of both how well this works in cells, but also how well this potential medicine works in animals. A really good understanding of the pharmacokinetics, which is when we administer this to a patient, we administer it directly into their spinal fluid. How long does it remain present in the nervous system? And so therefore, how frequently do we need to be administering this medicine for it to be effective? What are the levels where we see it to be active when we give it to an animal, and how do those compare to the levels where we might see toxicity, so that we can really choose what the appropriate doses are? And so those are the kinds of things that are critical for us to understand before we go into humans. And from the moment that we had this insight 5 years ago, we set ourselves the goal of trying to get this into patients as quickly as we possibly could, but not before we had enough of an understanding of all of those different parameters, so that we could be doing it in a way that we felt was safe and gave it the greatest likelihood of being effective.
Amazing. Neil, do your therapeutics have a somewhat different path? Maybe you have some thoughts on that.
Yeah, I have a lot of thoughts, but also I just wanted to make a point about the precision too, which is that precision also comes at at another level, and at the level of the specific therapeutic, as in the case of the ASOs. Of course, you achieve precision and specificity based on sequence. In the case of VectorY's program, they have a vectorized antibody that is selectively going after misfolded um, um, features, misfolded forms of TDP-43, and each of these new modalities as they come along, including CRISPR-Cas and other bioengineering technologies, are allowing increasing precision, that's allowing for us to be much more focused in our approaches. And there's a wonderful confluence, I think, now between an understanding of the biology of ALS and these new evolving technologies that's really driving the precision medicine approaches, not just in ALS, but across diseases. In terms of the path to the clinic, yeah, you're right. It's very different for the ultra rare than for a therapeutic that's intended to treat large numbers of ALS patients, sporadic patients. When I started the first program, there was very little experience with this. There was one other program that I know of, Tim Muir's Millicent program, where an N of 1 ASO had been introduced into the clinic. We learned a lot from his experience and built the J-Suffusin FUS-ASO program, but there were a lot of restrictions on us at the time, regulatory restrictions that don't exist now, because the FDA learned with us as we moved, these ultra-rare gene-based therapeutics forward. And in 2021, a couple of years after we initiated the FUS program, the FDA issued a series of guidance documents that now governs the development of ASOs for life-threatening, ultra-rare genetic diseases like the ALS cases that we work with. So now, for me, the restrictions, the regulatory restrictions to move into an ultra-rare case forward is much less than what Olga and Eric or anybody else working in a commercial program have to face. And so that is, based on an understanding that, if we were to apply the same kind of limits and restrictions, regulatory restrictions to the programs of the ultra rare, these people would die waiting for their individualized therapeutic to be developed. So there was an urgency that came from a need, a clinical need, and we have benefited from this. This is why we have been able, in just a few years, to get approval for, I don't know what we have now, probably a dozen INDs in the Silence ALS program. And we have an opportunity to move many more of these forward quickly. What we're learning, again, from these programs will apply, and I hope programs like Eric's and other companies who are developing ASOs with similar chemistries, similar technologies, will benefit from our experience in the ultra rare.
Thank you so much, Olga and Amy. Anything to add?
Yeah, maybe just a couple of thoughts, specific to our treatment modality. Obviously, it takes much longer. As Neil said, it cannot be as fast track as highly individualized ASOs for rare genetic subtypes, but for multiple reasons and for good reasons, our treatment is one and done. And it is the beauty of this treatment, but obviously we have to be very careful with the safety profile because we don't want to introduce any irreversible safety signals neither. So the preclinical program has to be designed to de-risk as much as possible any potential safety signals, but also to be very sophisticated in terms of how to select the clinical dose. Because with the treatment modalities like ours, we do not have the possibility to do classical dose finding in human patients. We have to start with at least minimally efficacious dose, and at the same time to have a sufficient safety coverage. So it gets very complicated. And then on the top of that, our route of administration is intracisterna magna. And so we need to test this clinical route of administration in non-human primates, also to de-risk the procedure, not only the safety of the drug. So I think every treatment modality, and also depending on the patient population, have their specifics. But at the end of the day, there are also a lot of similarities. We all want to go fast because the unmet medical need is huge, but we can't go faster than the evidence allows because the patient safety must come first. And I think this is our number one priority. And I think this is, the regulators are also on the same page, and it always comes at the end of the day to the benefit risk profile, so that we can be as fast as possible, but also as safe as possible.
That was beautifully stated, Olga. And Amy.
Yes, thanks, Nadia. I just add a couple of comments. One is that I think for now, there's a biomarker element to this, right? And so Neil has the benefit of being able to rely on genetic biomarkers, and that's an important part of patient inclusion. That's how you know who you're going to dose with your individualized ASO. It may not be required for every program right now, but I think more and more in the future, that's what biotech and pharma are striving for, to have that upfront biomarker that tells you, is this the right patient to include in my trial? And so this is part of the reason why Target ALS has developed this longitudinal biofluid core, to provide samples to allow them to do that biomarker work and have their biomarker assays in place for that clinical trial. The second piece is really around when you get into more individualized therapy, you're talking about smaller patient populations. And so where do those patients come from? Where do they live? And how can we funnel them to the right trials in the countries where they're being run? And so that's part of our goal is, through our global natural history study, is to work with underrepresented patient populations around the world, including Malaysia and India and other places in the global south, to connect them to connect these rare patient populations to biotech who may have a trial that's right for them.
It's really insightful, Amy. Thank you so much. All right, I want to make sure that we leave enough time for questions in our last 15 minutes. So we were going to dive into a little bit about how do you match the therapeutic type? How did you decide ASO versus intrabody? And so we'll do that, but we're going to do it kind of quickly here so that we get to biomarkers as well. So Olga, maybe we'll start with you. Your therapeutics, very interesting. How did you decide? How did VectorY come up with this? The design of the intrabody and the AAV?
Well, I think the number one point was that we knew that we cannot knock down TDP-43 because that will lead really to a disaster and potentially may kill a patient. So we had to design our intrabody with very high specificity to pathologically aggregated forms, and to ensure that there is no binding to nuclear TDP-43. The second element is that why we have chosen this approach is because we know that the biology of TDP-43 and the pathological aggregates are within motor neurons. And unfortunately, traditional antibodies act extracellularly, and so there is very low likelihood that we get even to the target with this kind of antibodies. And finally, we wanted to design the treatment to be durable and one and done treatment, and to reach the target cells, to have as high of a biodistribution as possible. And this is why we have chosen adeno-associated viral vector technology, which allows to get inside the cell and then bring the genetic material to encode for the intrabody, which then binds with high specificity to the pathologically aggregated TDP-43. So depending on the target, just TDP-43 as a whole is not something you can knock down, or you need to get where the pathology is, and to be very sophisticated in the way how you target it. This is why we have chosen vectorized antibody strategy here.
This is a great explanation. And Eric, how did you land on an ASO approach for UNC13A?
Yeah, I mentioned earlier that our understanding of the biology of UNC13A built on a lot of precedent from other researchers, including some disparate work even going on in worms. And I would say the same thing is true about the work that we've done with antisense oligos, is that, once we understood that improper splicing and processing of the RNA was the key problem, we were able to take a solution that had already been applied in another disease area, which is spinal muscular atrophy, where they had scientists from Ionis and Biogen had developed an antisense oligonucleotide that could alter the splicing and processing of an RNA in that disease, and that is now resulted in an approved medicine that's been transformative for people with spinal muscular atrophy. And so we really built upon the successes and the learnings from that experience to say, can we take that same approach and that same modality and apply it to this new target where we understand that splicing is also the problem?
And Neil, why are ASOs a good modality to use for the therapeutics that you're developing?
Well, they're a good modality because they are so modular and adaptable, and they can be used to treat different mechanisms. ASOs can be used to knock down genes through RNase H-mediated degradation of transcripts, or as Eric is saying, as splice altering ASOs. They can, they're sequence specific, so they can be very, rapidly designed for specific targets, and are particularly well suited for the ultra rare populations that we're dealing with. But the thing, one of the reasons why we've only worked with ASO so far in part is because there is a history of ASO development, not just in ALS, but particularly in ALS. And the FDA has a degree of comfort with this chemistry, with this modality that allows us to move forward quickly in the way I described, with regulatory guidances that allow us, a short path, relatively short path to the clinic. ASOs are excellent, I think, modality to begin a therapeutic discovery program. What I often think and say is that we're in the proof of concept business, really. That my interest is to demonstrate, in the case of FUS, as in the case of tofersen, SOD1, that knocking down a protein of interest, gene protein of interest, has a beneficial effect not only on biomarkers like NFL, but on the clinical course of disease. We know that's true for SOD1 tofersen. We know, I believe it's true for ulefnersen in the case of FUS ALS. I think it's going to be true in other forms of the disease as well. Once we have that proof of concept, others can go to work to try to develop better therapeutics. the one and dones, the more lasting, durable therapeutics, ones that perhaps have greater potency and efficacy, and that will be built on our work, demonstrating benefit with the ASOs. So I think we will probably begin to apply other modalities in our platform, but for the moment, the ASOs really are a great choice.
Thank you so much. And building on talking a little bit about neurofilament, Amy, we've seen a lot of discussion about neurofilament light and biomarkers in ALS because of tofersen. Are you seeing a shift towards more predictive markers? Who's going to benefit from a treatment? Are we getting in that direction in the science?
Thanks, Nadia. So I think what we're seeing is at the moment a lot of effort still, I would say, coming to fruition around trying to develop really sensitive and specific assays, both for TDP-43 itself, and people are taking novel methodological approaches to do that, to look for that in the plasma or CSF, as well as what's downstream from that TDP-43 mislocalization, meaning the cryptic exons or these altered RNAs like Eric is working on, like UNC13 and Stathman-2. So there's quite a big effort across the field on developing biofluid biomarkers to measure those things. And one of the benefits of the cryptics is that you could potentially not just determine whether you've altered TDP-43 itself, but rather, did you correct the downstream consequences and downstream function in terms of all of those different genes that are being misregulated at the level of the RNA? Can you see some potential correction there and then potential benefit on motor neuron degeneration? And so I see that there is some progress there using very specific kinds of ultrasensitive assays like Alimar's new ELISA assay. But I also see that there's quite a bit of work to go. This has been it's been a challenge for the field for some time to measure quite the right form of TDP-43. That's a pathological form, and that's robustly different in ALS versus control. And so there's some breakthroughs in the EV space there with Neurodex's work that they're doing, but again, still some work to go on those kinds of biomarkers, which are both what I would say both can be diagnostic biomarkers as well as treatment response biomarkers. So in the case of Trace, they can use an UNC13 cryptic peptide or cryptic exon biomarker should they be able to develop that assay in that space. But going beyond that, I would say we have been distributing biofluids from our natural history study, and also which are also accompanied by multimodal data sets, not just neurofilament light, but going beyond that to look more broadly at what are all the potential proteins that are changing in people with ALS and over time. So we can track those markers with disease progression or different disease subtypes like the C9 subtype, for example. So from that work, we're starting to see for sure some other additional potential treatment biomarker response biomarkers emerge, specifically a class of muscle-related proteins. And again, I think it's the tip of the iceberg for that work, with a lot more I expect to see over the next year emerging from both our data and samples, as well as work from other labs.
It's amazing. And Eric, can you talk a little bit about biomarker development for your UNC13A program?
Yeah, biomarkers are an area that we've placed a huge amount of emphasis over the past several years in parallel with the work that we've been doing to develop our potential therapeutic, developing the suite of biomarkers that's going to help us to understand as much as possible the effects of our medicine in people with ALS, and we hope give us the greatest confidence in the benefit that we may be able to see with it. And the way that we think about this is to try to develop a set of biomarkers that give us the full cascade from the immediate molecular effects of our therapy. So the kinds of things like Amy is talking about, where we can directly look at the presence of the UNC13A cryptic exon from patients' biofluids, and then starting to step forward from that molecular activity all the way through to the functional benefit we hope to see. So we'll be looking at biomarkers of the activity of the circuits that are being controlled by the synaptic communication that's regulated by UNC13A. So we have some electrophysiologic markers looking directly at that. We'll then be looking at the effects of those circuits on the muscles they're connected to. So Amy alluded to some of the emerging work on muscle proteins and muscle biomarkers that we're looking closely at. And then beyond these kinds of molecular biomarkers, ultimately what we all care about is improving patients' function and survival. And so we've been very interested in how we can use some emerging ultrasensitive digital and wearable tools that can give us a better sense of patient function, either in their movement or in their speech. And so all of these are things that we're incorporating as part of our clinical trials, together with some of the more conventional clinical scales that people have used in the past.
It's amazing to hear about the spectrum of different biomarkers that are involved here. And Olga, what types of biomarkers are you evaluating in your study?
I think like for Trace, biomarker development is really a top priority for us, and we have invested very early in this effort, not only in terms of having a set of biomarkers, but also to better understand the natural history trajectories of some of these novel biomarkers. We don't yet fully understand actually all of this and how they correlate over time. And this is why the work which is being done by Target ALS and helping the field to get access to biofluids and natural history studies done by Target and others are so helpful to the field. And for instance, we have partnered with an academic center, with Utrecht Medical Center, to conduct an observational study ahead of our clinical trial, where we enrolled patients who closely match in terms of their phenotype and disease stage, and collect biofluids from them to be able to better understand the trajectory of the TDP-43 related biomarkers, cryptic peptides, and obviously more conventional biomarkers such as NFL, and how this correlates with the novel biomarkers. So this is something we have been doing for a couple of last years. And of course, our treatment acts within the cell, and we cannot obviously take live more endurance from patients. So we have to rely on the proxy fluids. We have to do the CSF sampling to measure some of the target engagement biomarkers and downstream biomarkers. But again, I think there is no biomarker which, or a marker, any measure which on itself can help us understand how our treatment works. We have to rely on several complementary measures, like Eric mentioned, biomarkers, but also clinical outcomes. Obviously, I think the efforts everybody's doing, we have to share what we do and others do, so that the field can get stronger, because the biomarker field is really the key priority for ALS, I think, clinical development.
Yeah, thank you so much for sharing all of that. All right, I know that there was a lot of information here. We're gonna, at North Star, break some of these things down and share them in smaller clips so that people can really digest this information, because it's absolutely incredible. We're gonna shift to taking some questions that were pre-submitted, and also questions that we have in chat. So we have Lynn Brellmeyer, who's been living with ALS for 7 years, asking, many people are coming to the conclusion that ALS is more than one thing, and we may need multiple compounds, as they're used in oncology. So what other ideas would be complementary to the approaches that you have? And, feel free to jump in and share your thoughts.
I would agree that ALS is more than one thing. I mean, certainly there is a great deal of what we call phenotypic or clinical heterogeneity. No two ALS patients really look alike. And the variety, comes in age of onset and site of onset and rate of progression and degrees of upper versus lower motor neuron involvement and all kinds of, issues that determine, a patient's course. And, but while I think there is a great deal of difference between them, I think we can't lose uh, sight of the fact that there's a lot that's similar, and I would bet more that's similar than is different between these different forms of motor neuron disease, ALS, MND. And of course, as best we can, we want to target those shared mechanisms at common biology with therapeutics that can help as many people as possible. I think yeah, I guess, I mean, that's maybe somebody else wants to jump in, but
I can jump in too, Neil. Is that okay?
go ahead, yeah.
Okay, and maybe I'll let Olga and Eric also jump in. If I may, I'll just suggest that I think there are combination therapies that we're thinking about already. So, for example, combining Olga's treatment with Trace's VectorY's treatment with Trace's treatment might be a nice approach for patients where perhaps what patients need to understand is that every neuron is degenerating at a different rate. So they're not all at the same place at the same time in this, in the brain and the spinal cord. So having a combined therapy where you're treating the utmost root cause of TDP-43, but then you're also targeting at least one of the key downstream effects on 13 missplicing can be a really nice combination. So I think the field is certainly thinking about that. And last thing I'll mention is that we fund groups like UNC13A, which is a new evolving biotech, which has developed a specific modality or specific drug that combines the benefits of an ASO with the benefits of an siRNA into one single molecule that can allow us to tackle, let's say, proteins that are involved with motor neurodegeneration, like SAFMN2, along with proteins that are perhaps upstream, like TDP-43.
Yeah, Amy, I would agree. I think we have also a great example in the field with spinal muscular atrophy, where at the end of the day, most of the patients are on combination treatments. They would start with a gene therapy and then continue with an ASO, vice versa, or add a splicing oral modulator. So I think hopefully we will get to the stage. I think what we need to do now is to have the proof that these treatments work, improve our clinical trial designs, and improve the biomarker readouts. And then I think we will get to the point when we will have combo treatments. And I think this is valid for most of the neurodegenerative diseases in general.
Yeah, I'll just add, I think in the vast majority of cases, it's clear that ALS is a complex disease. There are multiple different pathologies that are important and that are occurring at the same time. And, I often hearken back to my own clinical trainings. My clinical training is as a cardiologist, and essentially every one of the common cardiovascular diseases we treat, we have gotten to effective treatments by combining a number of effective medicines over time, whether this be blood pressure or heart failure or cholesterol. And, my hope is that this will eventually be the story in ALS, and that we have to start by identifying individual nodes and targets and medicines that are effective, and that over time we can build these up together into even more effective combinations. And I think it's, from where we stand today, where we're really very much at the beginning of that journey, it's part of why I think, this community is one where we do all kind of root for each other, because, we're so early. And I think ultimately the best solutions here are going to be our ability to combine multiple effective therapies.
And we focused a lot today on mechanisms that are what I call intrinsic to the neuron. But there's ALS is a multicellular, multi-system disorder, as you're hearing, and there's a lot of work going on to develop therapeutics, for example, that focus on the inflammatory or immune aspects of the disease. And some of those may, in fact, be triggered by some of the nerve intrinsic, motor neuron intrinsic disorders. But we, it makes sense as we think about this that we would need not only to deal with the primary drivers of neuronal degeneration, but the secondary consequences, some of them which may be immune or inflammatory, that result from that early insult. So there's a very, I think, clear example of how therapeutic approaches can be combined to greater benefit.
Amazing. And I think we have time for one more question here. So this was a pre-submitted question, but people were asking what milestones they might look for from your programs in the next 2 years or so. So would love to hear from all of you. Olga, maybe we can start with you.
Well, as I said in the beginning, we are actively recruiting in our trial. So in terms of milestones, I would hope to complete enrollment and start getting the interim readouts. So this will be the next pretty much near-term milestones. So fingers crossed we deliver on this.
And Eric.
Yeah, so we, as I mentioned earlier, started clinical trials with our UNC13A targeting ASO earlier this summer, and expect to be having clinical data that we'll be sharing throughout the course of 2027. So we expect to be really exciting next 12 to 18 months for us.
Amazing. And Neil.
Yeah, a lot coming up. As I said, the first trial will read out later this year. Top line data is expected as early as October. I'm planning to go to meetings to talk about that data. I hope it's a victory lap. I don't know. We'll see. But it's a very exciting moment. I think for us who've been involved in this program and for the field, in the Silence cases, we have data. We have 4 or 5 ASOs now in the clinic. Some programs more mature than others. I hope that we can begin to show through a series of successes that this is an approach that works. This is a platform approach. This is a strategy that can be applied in an iterative way to begin to really make a dent in this disease. It's a very exciting moment, and I hope that the next even year or two will give us a lot to be happy about.
Amazing. And Amy, what should we see from Target ALS, do you think, in the next couple of years?
Yeah, thanks, Nadia. So for sure, we've seen recently that ALS did hit a milestone with their phase 2 proof of concept study showing that they were able to lower neurofilament heavy as well as neurofilament light, and had shown some clinical benefit as well. So we look forward to seeing the next milestone for them from their phase 3 program. And I don't know the timetables for that, but then of course we're very excited for VectorY and Trace to read out over the next year. And finally, there are a number of other biotech that we support in a number of ways. NRG Therapeutics is one, and we have a number of others in the Bay Area as well that we're supporting who should be entering phase 1 soon. So I'm really excited to see this class, this group of molecules going into clinic. I think we'll learn a lot from them.
Amazing. All right, we have a very nice comment for all of you. Thank you all for sharing such amazing info. The statement I wrote down that really resonated with me was from Olga. How close can we get to the root cause? That is so meaningful and can really help put context around how we in the patient community go about understanding the impact of research. You guys were all just amazing. Thank you so much for taking time out of your day to talk with us, to talk with the community. We look forward to continuing to sharing what we discussed today for months to come, because again, it was a lot to digest. And we can't thank you enough for all you're doing to advance ALS research.
Thank you
so much.
Have a great rest of your day, everyone. Take care.
Bye.